And I think I do somewhere. Aha. Everybody on this page. Okay. Has anybody been playing around with it at all?
Oh, they added out of school now.
No? Did you finally got internet, right?
No.
No, I thought you did. You said you did. No, it was Balboa got internet.
Yeah.
Oh, okay. All right. Let's do kind of what we did last time. Well, first off, let me do this meta-analysis. I got to get rid of this stuff. Hold on. Move all bosses. There we go. Okay. So let's go to, here's what I want you to do is I want you to put in breast cancer. Breast cancer. And here's what's going to happen is this phenotype thing is going to show up. Just click that first one. It says breast cancer SNOMED CT. Click it. Yep. That's just a kind of a classification, general classification they've already kind of assigned to this. Again, remember phenotypes are kind of a condition. So whatever condition. Did everybody, when you look at genetic markers, did this come up? Most correlated genes. Okay. All right, you guys with me? Balboa, are you on next bio? Okay, so we're at the next bio summary page, right? Click on these genetic markers. Yep. Is it coming? We might all hit the internet at the same time. Did anybody get any genes? Is it coming up?
Yeah, I got CCD1.
Yeah, that's the number one. Ah, there it is. Okay, does everybody, did it come up? Okay. This is cool. Basically what they have done, and you didn't even have to do any like big time computational work. What this did is it basically, based on their own algorithms, goes through and says, okay, here are all the breast cancer studies. I'm going to look for any genes that seems to basically, be significant or different in any of these studies. Given here is the name of the gene, and here are the supporting data types. So this first one is DNA methylation. What happens when DNA gets methylated? Remember? Do you remember what I told you? I think it was on Monday. I can't remember. I think it was Monday. Yeah, it was Monday.
I was here.
Oh, you were.
Monday?
Yeah. You weren't? Yes, you were. Remember, it was the first part of this presentation we did. So remember, DNA gets packed. What happens when DNA gets methylated? Do you remember? Okay. When DNA gets methylated, I only said it once. When DNA gets methylated, basically it gets more condensed. As the DNA gets more condensed, it means it starts shutting off genes. Right? You also have RNA expression, so that's basically mRNAs. We're measuring the emails that are coming off the genes. GT, the SNP GWAS. So basically what they've done is they know that in families that have breast cancer, this gene, mutant copy of this gene, typically gets inherited with the individuals that are affected. What is this? Copy number, CN. This is basically saying that sometimes they'll find breast cancer and this gene either gets duplicated or deleted. So normally you always have two copies. In this study, it's saying that actually you get more than one copy. And then again, this gene can be mutated. Put your cursor over this CCND1. What is this gene called? If you look down, so you'll see the symbol here, cyclin D1. And then you look at phenotypes, look at all the different cancers it's been associated with. Does everybody see that? Is everybody like scrolling up and down?
Yeah.
This is kind of cool because this can give you just a brief kind of summary on what it does. Proteins, cyclin family, function as regulators of CDK kinases. Function as a regulatory subunit required for cell cycle G1 to S transition. That last one, poli kinase, was G2 to M. Again, probably similar functions, but different cancers, maybe a different mechanism on how to basically proliferate the cells. But again, you can go in here and basically look at anything you want on this gene. If I click this entries gene, click on that one. Another window should pop up. Maybe. Did anybody else's pop up? Okay. Maybe if I pet my computer, it'll work better. There it is. This website that just popped up, this is NCBI, the National Center of Biological Information. I think that's what it stands for. I can't remember. This will give you all kinds of information, great information on whatever gene you're looking at. As we scroll down here, this will be a good page for you to go back. Here's what I want you to do is, why don't you bookmark this page? You will need it. All right, I'm gonna shut this. But again, we can read down here and you can see, like, you know, it's involved in cell cycle. That's a perfect expected process to be associated with cancer. We can go down here and it gives you, this is just other information. Here's a picture of our gene. Does everybody see this? These green bars are actually the only part, the gene goes from here to here, but it's only these dark green parts that actually get made into a protein. Those are called exons. The part in the middle are called introns. Exons are the only thing that actually code for the protein. And again, you can go down here and basically you can read about this gene, some of the papers it's been in. And as you can see, kinase independent role of cyclin D1 and chromosomal instability in mammary tumorigenesis. Right? We're looking at breast cancer, mammary tumorigenesis. Bam. Not only have other people seen that this gene is associated with breast cancer, but as I read this, I'm thinking it's an oncogene, for sure. This is where you're going to get your information, though. This is going to be kind of, you know, as you go through these lists, this is, you know, where you're going to get your information. So go back to next bio. Here's what we can do is when you get to this list, and this list isn't too big, right? Here's what I want you to do is as you start making these lists, I want you to export these results. Everybody has that button, export results? Tell me you do. Hit that. Is it exporting yet? Okay. When it finally does get to that point, basically just save it somewhere on your computer. Here's what I want you to do is I want you to start making some of these lists. Make sure you, whatever you do when you finally get to that point, label it something that you will know where it came from. Like this list will be genetic markers for breast cancer. So I'm going to go back to curated studies. Just let me know when your thing finally pops up. Oh, is it locked? Nope. It's not locked. Now, as we look here, so I'm at curated studies. So basically, this is telling me all the studies, the biological studies that this database has that is associated with breast cancer. How many studies are there?
Where that? Oh,
943 studies. That's pretty good eyes over there, oh my. Oh, no, he was looking up there. Here's the deal. That's way too many just to kind of peruse through. So here's what we can do is we can actually, let's start filtering this stuff. So I can go to organisms. Say I just want to look at human studies. I can click on that. Apply filter. Data types. Say I just want to look at genes that were associated with, say, RNA expression. I can click on that one. Apply filter. So we're whittling it away. Now there's 693 studies. Now here's where the keyword comes into play. Let's do this. What did we say? Was it stage, or did they call it class in the? What were they? The stage 1, 2 versus stage 3, right? Let's put stage and see what comes up. Or wait, class. Go class. And go class 2. See what that does. Hit return. Or apply filter. Now there's 20 studies. Did everybody get 20 studies? Please tell me you did. You got it? All right. So here's what I want you to do for the rest of the class period. So now we got studies. Look here. These are basically these different studies. All of these, some of these are $1 million studies that people basically created and then deposited the data that we can now look at for free. So we're looking at millions of dollars worth of research here. Click on the first one. Oh, there's no class there, is there?
You got 20.
You got 20? Sweet.
Almost.
Okay, I was looking at... Oh, it's called something else. Hold on a sec. I'm sorry. It's not doing stage, is it? Class. Wait a minute. Try this. Take out the class one. Do a keyword again. Now put in, let me try stage. Stage 2. Try stage 2. But you gotta cancel out that other one. So here's what I want you guys to do is, didn't really do anything, did it? Oh, here it is. Now what we can do, remember stage 3 is when the breast cancer gets critical, right? Go here, what I want you to do is find different studies that have just stage 3 versus something else. And you can add those to the meta-analysis. So if I click on that first one, there's a stage 3 versus stage 1. Look here. Any stages? Oh, grade. Oh, it's also grade too. Grade 3 versus grade 1. Stage 3 versus stage 1. So what you're doing is we're going through this data set and we're picking out all those genes that are different between stage 3 versus anything else. Here's another one. Do that, and then basically what you're going to do is you're going to end up in this meta-analysis. All these lists are going into this meta-analysis, this oil can looking thing. Here is how you generate your gene list. So what you're going to do is you're basically going to go to meta-analysis when you're done, and you're going to see all the lists that you have. Go to gene results and start downloading some of these genes. Basically, you're going to export this gene list. Oh, this is cool. Look at this. They're all going down. So this is the gene here. This is mRNA. It's saying this gene goes down in stage 3 versus everything else. This is probably what is it? Is it an oncogene or tumor suppressor? If it goes down as the cancer gets worse. What? Tumor suppressor. Good job. All right. And look, you just look at, honestly, if you start looking up some of these things, or even just looking at things, cell division, centromere, it makes sense. Okay, so till the end, here's what I want you to do is I want you to go through these curated data sets and start adding things to your meta-analysis using the stage thing. Oh, I just lost it. So remember, it's breast cancer. The first thing you do, you want humans. Data types, I would go RNA expression. And then keyword, I would say either stage, or what was that one they used, stage, and it was, what was that one? Stage, and then what was the, what, say it? Class? Oh yeah, maybe it was class. We'll try stage and see what happens.